White blood cells are important in defending your body from pathogens. The production of more white blood cells is going to be a result of infection. When you have lots of these pathogens in your body, that's going to release hormones that trigger the production of more white blood cells. Do recall that the myeloid stem cells that made red blood cells are also going to be kind of that precursor cell for basophils, eosinophils, neutrophils, and monocytes, which we'll talk about more. And the lymphoid stem cells really only become lymphocytes, your B cells and T cells. Now in lab this week, you're going to be looking at these different white blood cells under the microscope, but let's talk a little bit about what each of these do. So let's start with neutrophils. It's the most common white blood cell that you have. It makes up somewhere between 50 and 70% of your white blood cells. You'll see in these numbers there tends to be a lot of variation. So neutrophils are phagocytic cells, meaning that they engulf these foreign pathogens. Neutrophils are going to use some pretty gnarly chemicals. They use things like hydrogen peroxide, superoxide, some different enzymes, and a peptide called defensin. Hydrogen peroxide, superoxide, enzymes, and defensins. Right? These are pretty gnarly chemicals. So what ends up happening is a neutrophil really only survives engulfing 1 to 2 dozen bacteria before these chemicals are also going to destroy the neutrophil itself. But fortunately, we have a lot of them. Now, as far as identifying these in lab, typically you're going to look at things like color, the shape of the nucleus, and maybe the size of the cell. So just take a peek here at neutrophils. They have a very complex multiple-lobed nucleus. In fact, another name for neutrophil is polymorphonuclear cell, or polys, you'll hear them called sometime. So you're looking for that really complex nucleus, a cell that's bigger, maybe twice as big as a red blood cell, and really kind of the granules in the cytoplasm are pretty lightly stained. Okay, our eosinophils are this bright orange. So their granules actually pick up a pigment that's orange called eosin, right? They're twice the size of red blood cells, and they have this nucleus that has 2 lobes. So we say it's bilobed. Nucleus. So between that bright color and that bilobe nucleus, those should be easy to find in lab. Now functionally, eosinophils are also phagocytic cells, but they tend to go after multicellular parasites, organisms that are actually too big to be engulfed. And so they're going to produce some gnarly enzymes and chemicals, but they're really using exocytosis to put that out there and try to kill this multicellular or large pathogen. So they attack multicellular parasites. In fact, if someone, like a tapeworm or something, so someone who has a parasitic infection or allergies will have elevated levels of eosinophils. Otherwise, these are pretty rare, 2 to 4%. So that would mean you'd have to look through, 100 white blood cells to find 2 or 4 of these. Okay, multicellular parasites. These guys use enzymes and nitrous oxide, okay, to do that attack. Okay, the basophils. So basophils are small and stain really darkly. Here they're showing you a bilobed nucleus and these really dark staining granules. Under our microscopes, I don't think you'll even see the nucleus, but you will see a small, darkly stained white blood cell. These are really rare, less than 1% of your white blood cells, but they're super important. They contain histamine, which is important in inflammation, as well as heparin, which is an anticoagulant. Right. As we move on then, so all three of these have these granules that stained. They're actually called granulocytes. That's how they group them. Our last two, monocytes and lymphocytes, are considered agranulocytes, like without granules. And so let's look at our monocyte. Monocytes only stay in circulation about 24 hours, and then they're going to move out of the bloodstream and into tissues. When we do this, we change their name. Don't ask me why, not in charge. They change their name to macrophages. And so actually in the bloodstream, they're not that common, but throughout the body, you'll find them quite frequently. So monocytes are very distinctive under the microscope. They are large. Look how big this is. It's like the size of 3 red blood cells, and they have this very obvious kind of curved nucleus. Usually it's described as horseshoe-shaped. That kind of looks like a candy cane to me. It all depends on perspective, but it's pretty obvious. Now, monocytes or macrophages, these are some really important phagocytic cells. Right? So just like neutrophils, they're engulfing foreigners, but macrophages, monocytes, when they do this, they just keep going, right? They don't actually kind of burn themselves out in the process, and so they just keep eating and eating cells. Last but not least, are the lymphocytes, right? Lymphocytes are going to be part of our adaptive immune system. Everybody else, if it's foreign, we're attacking it, right? Lymphocytes are very picky. They need to have seen that particular strain of influenza or E. coli before in order to attack. So these lymphocytes, you may recognize B lymphocyte or B cell. Those produce antibodies. T cells, T lymphocytes or T cells, there's a huge variety. Some are kind of ramping up the immune system, some are simmering it down, and others will actually go out and attack infected cells themselves. We'll talk more about this for sure when we get into the immune system. As far as identifying lymphocytes, they're pretty small. Notice it's just a little bit bigger than that neighboring white red blood cell. And the other distinctive thing here is, notice this kind of ring, a really narrow ring of cytoplasm, almost like a little halo. That's very distinctive. What I see students confusing it with is basophils. They're like, oh, it's like small and dark. Look for that little ring of cytoplasm, and that'll be your lymphocyte. Lymphocytes are pretty common. You'll see a lot of them, so hopefully that will help you distinguish them. Now, the way we used to do a differential white blood cell count is you'd look at a microscope slide. You just go back and forth, systematically, not recounting any cell. You try to count 100 cells. And each time you see a monocyte, you're clicking a button. Each time you see a lymphocyte, right? So you're keeping track or tallying, keeping little tick marks. To see those percentages, right? If something falls outside of the norm, it can give you some clues as to the person's illness. Now, that was tedious, and so now we have equipment that a light will actually shine through a blood sample, and a computer picks up the resulting image and basically compares the complexity of the nucleus with the size of the cell. So a big cell with a non-complex nucleus is a monocyte. A very complex nucleus in a pretty good sized cell is a neutrophil, right? So see how that works? And then of course the computer can count the number of dots, and they'll actually print out for the healthcare provider what those percentages are. I know we have a slide, for example, in the lab of someone who has an elevated number of eosinophils. Again, parasitic infection or allergy, I don't know. but that shows up on a differential white blood cell count and is a really good clue that it is a parasite or an allergy. Okay.